Yeah, the story today is about Revel Pharmaceuticals and our hope to kind of repair some of the root causes, what we believe are root causes of aging and disease. I'm part of the California contingent of people that have come out here. It seems like there's quite a few of us, and looks like there's Aaron coming after me as well, who's also California-based, so it's great to have a lot of the Sunshine State representation. A little bit about my background to start. I came out of Stanford with a PhD a few years ago and had the opportunity to work at Google X and a few biotechs in the Bay Area. And I just, had these interesting job opportunities, but I felt this desire to kind of apply my energies and skills towards this interesting area of aging biology that I had read about many, many years ago in Aubrey de Grey's book. And so that's the story I'll be sharing today. So first, zooming out a little bit, over the last few days, I'm sure everyone has come to their own definition of what they view aging as. And I really like this definition, that aging is the accumulation of damage in molecules, cells, and tissues over a lifetime. And ultimately everything's made of molecules, so we could almost simplify this definition to aging is the accumulation of damage in molecules. And to give you a more visceral sense of what I mean by damage, I'm showing here two different tissue sections, one from a small artery of an 8-year-old and another from an artery of a 50-year-old. And the sections have been stained for a particular type of damage known as an advanced glycation end product, or AGE, or age. And the staining is brown. So as you might expect, the small artery from the 8-year-old, nice and clean, no brown staining, right? Unfortunately, most of us in the audience probably have arteries that look more like this 50-year-old, healthy 50-year-old, where there's this diffuse, staining throughout the tissue, but really intense staining kind of along the artery wall. And Revel's interest is in trying to reverse, basically, the staining so that the old tissue looks more like the young tissue from the 8-year-old. But I want to go one level further, because this is still a bit abstract from the level of molecules, and actually identify the chemical culprit of what we talk about when we say an advanced glycation end product or a damaged product. And this is what an AGE looks like. There's a few different AGEs that are prominent in the human body, but this is one example, an age known as CML, or carboxymethyl lysine. And basically, it's a lysine, as you can see on the left, that's been modified with a small sugar group, or sugar-derived group, that alters the property now of this chemical constituent in tissues. And everything in the body is made of molecules. DNA is a molecule. If you change the nature of that molecule, it no longer functions the same, and it may have inflammatory properties, or the properties are just altered. And so this small little change actually makes a big impact. And so the mission of Revel and kind of our vision and purpose for existence is really to develop therapies that are capable of repairing these molecular damage products. So repairing age-related damage at the molecular level. So that's the talk. That's all I have. You can go to sleep if you're ready. Otherwise, I have a little bit more startup work. But as long as you remember what I just talked about, that's the message. And I think it's often lost because we think about cells and tissues, but ultimately everything is constituted of molecules. So condensing what I just said as a little startup pitch, basically many diseases are caused by dysfunctional accumulation of toxic proteins metabolites at the molecular level. Revel exists to develop enzymes that can reverse this, reverse the damage, hopefully reverse the course of disease. And so to this end, we've developed an enzyme engineering platform that allows us to create the biocatalyst that can convert a damaged product back to its original form. So hopefully you've heard of enzymes. You may have had enzymes injected into your face if you've ever used Botox or Dysport. Enzymes are probably in your laundry detergents at home. They're used to efficiently remove lipids or fats and protein stains from clothing. And enzymes are used commercially to help manufacture pharmaceuticals. So it's a robust, heavily utilized class of molecules. And we've developed an enzyme engineering platform that allows us to find and identify and improve enzymes that can carry out repair-based catalysis. And we're applying this platform to quite a few different damaged products. So I've listed a few here. These are just chemical names of things we're trying to repair. So citrulline, carbamylation, CML, the AGE I was talking about earlier, all just chemical names. And basically, I think like many of the sister companies here today are kind of have been discussing, we have a conventional biotech arm where we've found a damaged product, kind of matched it with a disease that seems promising for conventional biotech investors, and there's biology supporting that. But ultimately, we hope that we can translate conventional approvals into longevity biotech, because the damaged products shown here do accumulate with aging and have universally throughout our bodies overall accumulation over time. Briefly, just a little bit about some of the markets we're looking at. Citrulline is a really interesting product. We're probably going to be building our Series A around that one. It focused on rheumatoid arthritis. So if you have anyone in your life that has RA, you may have heard of this ACPA-positive class of RA patients. It'd be great to talk to you after my talk to just hear about your experiences with rheumatoid arthritis drugs and how it's affected you and how we hopefully will be able to help in the future. And then we have a few other conventional biotech plays based on just mechanistic mechanisms of actions for these damaged products. Ultimately, the longevity biotech market is the one that's really exciting, right? I don't think anyone would disagree. It's a successful molecule that can treat many conditions of aging will have a huge total addressable market. So for the rest of the talk, I'm going to dive in a little bit deeper into one of our advanced glycation end products, kind of share some of the exciting work we've been building over the last few years. And then I'll zoom back out at the very end, briefly talk about pipeline again and kind of our vision. So CML is a toxic advanced glycation end product. It accumulates to high levels over time. When at age 50, you might have X amount. At age 55, you're now going to have X plus some additional. It will only go up until you die. And it comes in two forms. So there's free CML that's just floating in your blood. And for the most part, if you have healthy kidney function, that will not build up in the blood. It will get excreted as any other toxin would by the kidney. And then there's bound CML. And this one's not going anywhere. So, once it accumulates, for the most part, it's just going to build up in your tissues. And, these are problematic for various reasons. Free CML has been shown to contribute to microglial dysfunction. There's some systemic vascular inflammation as a result of CML levels in blood. But the protein-bound CML is really the one, in terms of aging focus. It's involved in biological aging of the ECM. There's inflammation, systemic inflammation via activation of some inflammatory receptors that are dedicated to AGEs. And then it's altering the molecular constituents of your tissues in a way that biology didn't plan for, right? The age 70 you wasn't planned for in evolution in terms of these damaged products that have built up. So at Revel, we've developed enzymes that repair CML damage. So over time, you get modification, aging, and disease. And with enzymatic repair, we can kind of lawnmower off these modifications. And how are we doing? So with the free CML, which is the easier target because it's floating freely, it's not bound, and kind of difficult to access, we started off, it was rough. We didn't have much activity. And over the last few years, we've managed to improve the activity of these enzymes by a millionfold. So this is a relatively new field in terms of repairing these things. People have known about AGEs for more than 40 years. There's just been kind of no good attempts to actually remove them or reduce them. So this is really exciting data. First time we're really showing this. And we've actually exceeded a threshold for having useful activity on free CML in blood. And our enzymes are active in blood. They're stable for more than the half-life that they would have in circulation. So these are actually, effectively the problem of clearing free CML from blood is a solved problem. But the kidneys already kind of solve that, right? So if you have healthy kidney function, CML is not going to be accumulating in circulation. It's going to be filtered out. But the reason I'm sharing this with you is because we view free CML as a stepping stone towards the more challenging target of protein-bound CML. And this, I really would like to emphasize, these results are quite cutting edge. I think the field of AGE biology, we weren't even sure if it's possible to repair and remove these things from proteins. So very complicated substrates. There's not any enzymes that exist naturally that can carry out this reaction of bound CML. So yeah, we're making a lot of progress. Since 2019, we had 0 activity to start with, and now we've clawed our way up towards something that's looking to be useful, a useful amount of activity on tissue-bound CML. We're not quite there, but given the rate of improvement, we should be there in the next few quarters. So where this is going, actually I'll be sharing that on the next slide. One more piece of data. Because of the interesting kind of uniqueness of what we're doing, we've been able to engage with a biotech collaborator. They've been generating some data with one of our enzymes showing reversal or corroborating reversal of CML damage across multiple sites. So they've basically done some proteomics for us, showing, this graph is showing different peptide sequences that are constituents of a larger protein. And the red boxes are just kind of highlighting, a repair event. So a high blue bar basically means there's been a repair event at that particular site in the protein. And this corresponding protein structure is just kind of showing where those repair events are taking place. So again, yeah, these are results that I think the field wasn't sure if it was even possible to repair, intact proteins, and we've made a lot of headway in the last 4 or 5 years. So what's next? What's the kind of big vision? Well, as I kind of started the talk, right, you can imagine where this is going. We basically want these enzymes to be able to lawnmower out these damaged products in tissues, right? So one of the key experiments we're getting ready to run is taking human tissue sections from a 70-year-old person or having various diseases and basically showing we can lawnmower away with an enzyme these damaged products and repair the tissue back to its original molecular kind of constituents, at least for a particular product or type of damage. So some of our results from our biotech collaboration will be published over the summer or late fall, and we'll probably reveal who we've been working with at that time. But for now, I'm going to zoom back out to kind of our overall pipeline strategy. So one of the key things in terms of which damage targets we've been interested in pursuing has been which ones will allow us to get to clinic as quickly as possible. And there's some really interesting damage products like glucosapane. I've thrown lipofuscin on here, although we're not actively working on it yet, that are very technically challenging to pursue. So we've chosen damage products that we know we can kind of hit and be able to remove a little bit more easily on the technical side. Build a team, build a pipeline around that for engineering, so that later, when we want to pursue these more challenging targets, we have that in place and can just kind of plug and chug, hopefully. We hope to obtain clinical approvals, right, to validate the repair-based approach, whether it's us or one of the sister companies here. Anyone validating a repair-based therapeutic approach will be a huge turning point for the field, I think, right? Just showing that you can repair or kind of restore versus just trying to slow down and prevent is a really big conceptual change. And we hope, whether it's us or someone else, that will drive momentum into, our more challenging targets and the things the other companies are doing here as well. And, then expand those narrow clinical approvals towards aging and more general use. So briefly about our financing. We're actually not currently fundraising. Still would love to talk to investors, so if you'd like to email or meet up here, that would be great. We've raised about $9 million to date and been fortunate to receive about $1.2 million in grants from the NIH. These are actually pretty competitive. We have 4 of them, and they're like 20% success rate. So it's a nice scientific validation of kind of the quality of what we've been working on. With our current funding, we're looking to get to a development candidate and have a fully developed TCP and TPP for our product before Series A. So that's kind of our trigger. If needed, we'll move up our Series A. If we have this data sooner, we might push it back a little bit if we need more time to collect the data. But this is the core package we're really looking to raise around. And then the fund use. So ultimately, Series A fund use, collectively, we're hoping to get to a phase 1 patient dosing with that fundraise, or maybe a little bit after. So zooming back out in the final level, going back to the original definitions, I wanted to reflect upon just thinking about aging, right? It's important to define and think about what's actually happening. And a lot of it is happening at the molecular level, right? We talk about cells and tissues, but ultimately, it's the molecules that are changing. And so our view of rejuvenation is being able to repair accumulated damage at the molecular level and applying that to both molecule cells and tissues. So with that, thanks very much. Thanks for your attention, and happy to answer any questions.